in this study, again, as I said, we collect cerebrospinal fluid from MS patients and healthy controls, and we measure protein levels in cerebrospinal fluid. And we measure these protein levels by technology called SomaScan, which allows you to measure a lot of proteins in very small volume of cerebrospinal fluid. So in our approach, we measured 5,000 different analytes in this tiny amount of CSF. And our cohort of patients, of MS patients and healthy controls, is very well characterized. So we spend a lot of time with each of our patients that comes to NIH and is enrolled in our protocol, and we run several tests, we do neurological examination, and we have developed a lot of outcomes that measure the level of clinical disability and also outcomes that measure disease severity. And the difference between disability and severity is that the disability tells you the level of troubles that you are in as a patient, and the severity tells you how fast you are accumulating disability. So MS severity outcome can differentiate patients that accumulated different amount of disability over the same period of time. And what is good about MS severity outcomes is that if you find processes that are associated with MS severity, the chances are that many of these processes might be causal, might be causing the disease itself, right? So we have developed these outcomes, and in our patients, we have measures of clinical cognitive disability and how bad disease they have. On the other hand, we have protein levels of 5,000 proteins in cerebrospinal fluid. So we correlated these two entities and asked the question, which proteins are correlating with progression of patients and with severity? And then we used commercially available tool called Ingenuity Pathway Analysis. And this is a database of curated knowledge about biology of all sorts derived from human studies, animal studies, cell culture studies. And we asked the question, can we identify biology that is related to these biomarkers that are changing with how patients progress? And by doing this, we identified several pathways that are correlating with patients' progression. Again, we show that MS patients, as they progress, they are increasing the amount of neuroinflammation in their brain. We also show that as patients are progressing, they have activation of pathways related to fibrosis. Fibrosis is process of creating of fibrotic tissue. It's usually associated with wound healing. Let's say if you damage your skin, you will create a scar, which is fibrotic tissue. So similar process is happening in the brain of MS patients. As they have damage to their tissue, the fibrosis follows that is trying to patch up the damaged tissue. So neuroinflammation and fibrosis were the two big groups of pathways that we identified as being correlated with MS progression. The other quite interesting finding from our study is in MS, there is a really unexplained and unanswered paradox that some patients have predominantly disease of the brain, whereas other patients have predominant disease of the spinal cord. And nobody really understood what is the molecular basis of this difference. Is it just a stochastic process, or is there some molecular mechanism underlying this difference? And because we have all these different clinical outcomes that can measure clinical and clinical disability at the cognitive level and at the physical level, we were able to isolate outcome that measures the amount of spinal cord damage, the spinal cord disability. And that allowed us, again, to look at the CSF biomarkers and compare them between patients that have predominant involvement of spinal cord and patients that have predominant involvement of the brain. and ask the question, what is the biology underlying this difference? And what we found out was that patients that have predominantly brain damage have activation of neuroinflammatory pathways, whereas the patients that have involvement of the spinal cord as the main driver of the disease, they have more of the activation of fibrotic pathways. So that was quite new finding and interesting finding that might be used again in designing drugs for patients that have different localization of the disease in the central nervous system. And maybe the last thing that I would mention, all of these analyses that I described, they use the Ingenuity Pathway tool to identify the biology. But then another interesting and exciting outcome of our study was that we were able to generate machine learning models that are based off these CSF biomarkers that can predict outcomes of MS disability and severity. In other words, you can take 50 microliters of cerebrospinal fluid from any patient, MS patient. You run the SomaScan assay, get the results for these 5,000 biomarkers, and then plug these numbers into these models. And these models would predict what is the level of the amount of lesions in the brain, the amount of physical disability, cognitive disability, or MS severity, which on one hand is exciting because you can tell a lot about patient status just from a drop of cerebrospinal fluid. On the other hand, it would allow you to use data for patients that had cerebrospinal fluid collected, let's say at some point in the past, but they had no neuroexam associated with that visit, or they had no MRI associated with that visit. So the lack of the clinical outcomes that would characterize the phenotype of the patient at that time would be now solved by predicting the levels of these outcomes just from a drop of cerebrospinal fluid. That would also allow, creating consortia of centers that would be able to merge their CSF samples even if they are not having the associated outcomes with them.